In this video, we'll be starting with the chapter of DNA. Now, this chapter is found in paper 2 and it consists of around 27 marks. When you study, remember, have your exam guidelines with you always, as they will tell you exactly what you need to study so that you don't study any unnecessary work. Now, according to our exam guideline, we need to know the general cell structure with an emphasis on the ribosome, cytoplasm, and in certain parts of the nucleus. We need to know that there are two types of nucleic acids, DNA and RNA, and that they consist of nucleotides. Now, looking at the ribosome, these structures should not be, or these organelles should not be strange to you, as you've done quite a lot of this in grade 10. So this diagram on the left here is a general structure of a cell that you would find in an animal cell. You've got the basics like the mitochondria, the vacuoles, the centrosomes, all of those things that you would find in a cell. Now, looking at the ribosome, the function of the ribosome is that it is responsible for protein synthesis. Now, synthesis means to make or produce. So in this case, protein production, to make proteins. And the general structure of a ribosome is, as on this diagram on the right hand side, it has two units, a large subunit and then a smaller, so the smaller subunit, and then these two fit on top of each other like so, and then the proteins will be coming out at the end there. Then the cytoplasm, it's a jelly-like fluid mostly made up of salt and water, and this fills the cell and gives the cell its shape. Very important. Without the cytoplasm, a cell would probably look like a deflated raisin. And then it keeps the organelles in place. Then the nucleus, which is this structure over here, it stores the cell's hereditary material in the form of DNA, and then it also coordinates cell activities such as the protein synthesis, which we'll look at in a later video, and cell division. There are more activities. These are just some of the major ones. Then, looking at the structure of the nucleus, on this diagram on the right, you've got the endoplasmic reticulum, you've got the nucleolus. Let me get a different color. The nucleolus over there, and then you've got the nuclear membrane, which is a double membrane surrounding the nucleus. You've got the nucleoplasm on the inside, the chromatin, and then the nucleopore. So the nuclear membrane is porous to allow transport of material between the nucleus and the cytoplasm so that material can move in and out. Then the nucleoplasm is basically like the cytoplasm of the nucleus. It has a jelly-like fluid or liquid that fills it on the inside. Then the nucleolus, this round structure, it produces RNA and ribosomes, so this is where they are made. And then the chromatin network, this pool of noodle-like structures that you can see inside the nucleus, is a mass of genetic material that is composed of DNA and proteins that will form chromosomes if they clump together. Clump together is a bad word. Let's use the word condense. So if they condense, they'll form chromosomes. Next is the nucleic acids. So there are two types of nucleic acids that you need to know of, and that is DNA, which I'm sure you've heard of before, and DNA stands for deoxyribonucleic acid, and then the second one is RNA, ribonucleic acid. Now by the end we'll be able to make a table with the differences between the two, but just reading the name, the major difference comes in with the type of sugar. So ribonucleic acid uses ribose sugar, whereas DNA has deoxyribose sugar that is involved. Now, nucleic acids control protein synthesis. Once again, we'll look at that in a later video. Now, the general structure of a nucleic acid, it is made up of nucleotides. So these three structures you see on the right, all of them together makes up a nucleotide, which is the building block of these nucleic acids. So a nucleotide consists of a nitrogenous base, a sugar that is a pentose sugar, pent meaning 5, so a 5-carbon sugar, 3, 4, 5, and then a phosphate group attached to the sugar. So these three things make up a nitrogenous base, and we'll look at that in more detail just a bit later on in the video, especially with regards to the nitrogenous bases, because there's a rule of thumb that needs to be followed there. DNA structure and its functions. So the exam guidelines a lot of the time will already give you information that you can study just straight from the exam guidelines and not even worry about looking at your textbook. So in this case, they've already given you the location of DNA, where it is found. So you can find DNA in the nucleus, known as nuclear DNA. And then another type of DNA that will become quite important when we look at the chapter of evolution and even genetics and inheritance is mitochondrial DNA found in the mitochondria. We will look at a brief history of the discovery of DNA with Watson, Crick, Franklin, and Wilkins. Those 4 people are quite important. And then we'll look at the components of a DNA nucleotide in more detail, specifically the nitrogenous bases that are involved there. And then you need to know, once again giving you the study material in the exam guidelines, that the shape of a DNA molecule is a double helix. You need to be able to use or draw a stick diagram of the DNA molecule to illustrate its structure. And then lastly, they've given you the functions of DNA already. So basically DNA is there for hereditary information to be carried on, and then also the coded information for making proteins is found in DNA. DNA is quite a long history, but the discovery of DNA is attributed to these 4 scientists, and it is also a bit of a controversial one. There was lots of sneaky people moving around showing pictures that they shouldn't be showing to people that shouldn't be seeing it. And let's start off with identifying these 4 people. So this guy over here is James Watson, then Francis Crick, Rosalind Franklin, and then Maurice Wilkins. So Watson and Crick were already busy investigating DNA, but so was Rosalind Franklin. And in 1951, she started at King's College because she actually used X-ray techniques to study DNA structure. She wanted to understand what DNA looked like and what its components were, but so were Watson and Crick. So in 1952, Rosalind Franklin was able to take an X-ray photo of a chromosome. So this was groundbreaking stuff. This had never been done before. And it actually took her 100 hours to get this image on the right-hand side, which is known as photo 51, and it's a very famous photo. And this is a photo of a chromosome that she was able to take. And this was going to help her determine the structure of DNA just by looking at that picture. Now, by looking at that picture, it just looks like a X to me. But to them, it was quite vital information. But unbeknownst to Rosalind Franklin, Maurice Wilkins, who she wasn't really getting along with, took this picture and he went and he showed it to Watson and Crick. And this helped them greatly with their research, and looking at that picture they were able to come to a conclusion and formulate basically a little model that the DNA structure is a double helix structure, and that happened in 1953. So they released a paper just before Rosalind Franklin. It took her about a year to process all of the information she got from that photo. And she also released a scientific paper on the structure of DNA, and that came out just after theirs. and that made it look like she was just confirming what they had found, even though what she found helped them greatly. She passed away in 1958, just before Watson, Crick, and Wilkins won the Nobel Prize for Physiology and Medicine for the structure of DNA in 1962. So all of her hard work basically was never rewarded. And for those of you that are interested, this is what a Nobel Prize looks like, just for interest's sake. And then I did put a link to a video on the discovery of DNA in the description tab below if you're interested in looking at that video, because this is a very brief description of all the things that occur. Looking at the structure of DNA, it is a double helix shape. So the shape on the right hand side, it's all coiled up, and when it is uncoiled, it will look like a ladder, similar to this diagram on the right hand side. Now a DNA molecule, we've already discussed this, is made up of nucleotides, and these nucleotides are made up of a nitrogenous base, a phosphate group, and then a sugar group. And these nitrogenous bases will differ. So there are 4 nitrogenous bases associated with DNA. It is adenine, thymine, guanine, and cytosine. Now these nitrogenous bases can only bond with a certain nitrogenous base. So in the case of adenine, adenine can only bind with thymine, and guanine can only bind with cytosine. So cytosine will never bind with adenine or thymine, and adenine will never bind with guanine or cytosine, for example. And the way I remember which ones go where is always together is for adenine and thymine. They are always together, and then guanine and cytosine make a good couple. G for guanine, C for cytosine, good couple, A for adenine, T for thymine, always together. So they will always bond with each other in that specific order. Now, when you have to draw a stick diagram, this is basically what you will have to draw. So the nitrogenous base is attached to the sugar, which is attached to the phosphate group. Then the phosphate group will attach to the next nucleotide, which attaches to the sugar, the nitrogenous base, and then off of that sugar will be the next phosphate group. So you can see how these nucleotides are basically building this DNA structure. Now that is only one half of the DNA molecule, as the other half is on the right hand side, and how these two legs of the ladder basically come together is through weak hydrogen bonds. So the weak hydrogen bonds are found between the nitrogenous bases. That's how they are attached to each other. And the reason that they have to be weak hydrogen bonds is because at some point the DNA is going to be unzipped. So just like you unzip your jacket, it's going to unzip, and it's going to expose these nitrogenous bases so that they can be copied a bit later on during the process of DNA replication and protein synthesis. So they will unzip and then they are exposed. Because there are these different nitrogenous bases, the sequence in which they occur will give rise to certain proteins. For example, this could be guanine, then it can be adenine, thymine, cytosine, guanine. There's various combinations that can occur, hundreds and thousands of them, that will give rise to certain proteins. So that is the general structure of DNA. Now we can look at the process of DNA replication. So you need to know when in the cell it takes place, cell cycle it takes place, where in the cell it takes place, how this process occurs without knowing the names of the enzymes, so I won't even mention that in this explanation. And then also the significance. Why is it important for DNA replication to take place. So the process of DNA replication takes place in the nucleus during interphase of the cell cycle. And then the process itself, I have actually taken a memo from a previous exam paper where they asked to explain the process of DNA replication as well as the importance, so that you can see precisely where marks are allocated. So the process of DNA replication starts when the double-stranded DNA unwinds. So from that coiled form, it unwinds and it becomes that lattice structure. Then the next thing that will happen is that that lattice structure will unzip when those weak hydrogen bonds break. And then each of those DNA strands acts as a template to form a complementary DNA strand, and you can actually mention the nitrogenous bases, but in the form of the base pairs. So you need to mention that adenine will go with thymine and guanine with cytosine. And then using the free DNA nucleotides from the nucleoplasm, so those nucleotides that we looked at, so that nitrogenous base attached to the pentose sugar attached to the phosphate group, there is free floating nucleotides in the nucleoplasm. So those are the ones that will be used to form these complementary strands. And then two genetically identical DNA molecules are formed. So let's look at this diagram on the right here. So what will happen is this DNA molecule unwinds, and then it basically goes and lies in this ladder shape. What will then happen is the weak hydrogen bonds between the nitrogenous bases start to break, and the DNA unzips, much like it has here. And when that happens, these nitrogenous bases are now exposed. And because they're exposed, free-floating nucleotides, so these groups of 3, in the nucleoplasm come in, and they can try and find a partner for them. So in the case of guanine, a nucleotide with a cytosine will come and attach to it there. Adenine will have a thymine. This guanine is already bonded with a cytosine, and the thymine with the adenine, and so it carries on. And on this side, it will do the same. So this cytosine that was actually with this guanine will now bond with a free-floating guanine. This thymine that was with this adenine will bond with a free-floating adenine. And in the end, you'll have a complementary identical strand. So there are 2 identical strands of DNA that are then formed. And why does this have to happen? This comes in with the importance. So it ensures that each daughter cell gets an identical copy of the DNA after mitosis, and then it also ensures that each daughter cell gets the correct number of chromosomes after mitosis, which is very important. DNA also comes handy in different situations. In the case of DNA profiling or DNA fingerprinting, DNA is also used. So the definition of a DNA profile I also got from an old memo from a previous exam paper, and it is the process by which the DNA of a person or an organism is mapped, so it's laid out, or the DNA sequence of an individual is determined, or the barcode pattern of DNA is determined. So looking at these diagrams on the right, they look like barcodes, and that is the DNA sequence that is then determined for each person. Where do we use DNA profiling? So to investigate crimes, to resolve disputes, to solve murders, to identify organisms from their remains. So if there's nothing else except maybe a few particles of bones or tissue, you can use that to determine or identify the organisms, to identify missing persons, to identify family relationships other than paternity, for example, siblings or cousins, to test for the presence of specific alleles or genes that cause a genetic disorder, or to establish matching tissues for organ transplants. That last one is quite important. Now that we know the uses, you also need to know how to interpret these DNA profiles, and you will use it again in the chapter of genetics and inheritance when paternity testing is done. You need to be able to write about this in an essay even. Now, this diagram at the bottom here is generally the one that you will see in an exam paper, but let's look at this one at the top. It's a bit easier to look at for now. So this person was most likely murdered with this knife. Now on the knife you will find the DNA of the person that was murdered, and perhaps the person that committed the crime also left some DNA. Maybe they cut themselves accidentally, or there's maybe some skin cells or something that was left on the knife that they were able to take and extract. Now, because there are going to be two types of DNA on there, we first need to kind of rule out the victim's DNA on this murder weapon, so that we can just compare it with the three suspects that they found. So now we need to take out all of the corresponding barcodes of the victim on the knife, which means that would be her DNA. So let's cross out that bar and this one, and just I always have a ruler with me when I do this kind of thing, just so I know I'm crossing out the correct barcode. And just for interest's sake, just cross out the ones that correspond with the suspect as well, because it's not going to be that person. So there's a match. So this barcode is taken out of the account. Let's see. These two, this is why I use a ruler, so you can see that these two are not lying across from each other, so I won't cross those ones out. These ones we can cross out. Okay, so now we've cancelled all of the barcodes of the ones that could be a match to the female. So now let's see, the remaining DNA should then be of one of the suspects. Now we need to see which of these suspects' DNA matches the most with the weapon, the DNA found on the weapon. So here, let's see who that corresponds with. This one, this one, and this one. This one's a bit higher, so it doesn't correspond. The next one would be here, corresponds over there, and then lastly, now we need to go see which of the suspects has the most corresponding barcodes with the DNA found on the weapon. So suspect 1 has 2 corresponding barcodes, suspect number 2 has 3, and suspect number 3 is completely innocent. He has nothing. So in this case, the person that most likely committed the murder was suspect number 2. Now this can also be used to identify paternity. So same principle again, you're going to go look which of the barcodes correspond with the child's. So the mother and the child, remember you get DNA from your mother and your father. So let's just cancel out all of these. Okay, now we can start and we can see which person was most likely the father. So on this one, those two correspond, that one and that one corresponds, this one and this one corresponds. So now we've looked at all of the DNA. Now let's see which one corresponds the most. Male 1 has 1 corresponding bar, male 2 has 2, and male 3 has 3. So male 3 is the father of the child. And that is the end of the first DNA video.